We've talked about the way that human genetic disorders are inherited, but let's get into some specifics. We made some general references to these disorders already, but let's get into some detail here. So first of all, obviously we've established already that there's a link between genotype and phenotype, but now we have the capability to actually look at genes and see how that link is made. This is a really gross example, but people of African and European ancestry are more likely to have the dominant form of the earwax condition, which is wet earwax. So gross. People who are Asian or Native American have the dry form, which is recessive. The difference between these two phenotypes is a single DNA-based change. We'll get into next semester how the DNA, there are 4 letters in the DNA alphabet, and a gene can have hundreds, thousands of these letters in the protein, in the directions how to produce a protein. It's one letter that's changed from a G to an A. And that gene codes for a membrane transport protein. It causes the protein to produce dry earwax instead of wet earwax. One little change in that code. There's a direct connection between molecule and trait, and between genotype and phenotype. So there's a molecular basis for genetic disorders. Changes in the DNA that we've been calling mutations, there's the M word again, can change proteins by altering their amino acid sequences, and that can directly affect one's phenotype. There are thousands of genetic disorders that have been identified that are caused by mutations or changes in individual genes. Remember that the whole purpose of DNA in the nucleus, or not in a nucleus in the case of prokaryotes, is to produce proteins. So these changes affect specific proteins associated with cellular functions. You've heard of sickle cell disease already. We watched the video in class about Carlos Boozer's son, who was affected with a very severe form of sickle cell disease. This disorder is caused by a defective allele for beta globin. They're 2 different proteins or polypeptides in hemoglobin. Hemoglobin, recall, carries oxygen. That's what's in charge of carrying oxygen in red blood cells. So beta globin is not produced properly in the case of sickle cell disease. It makes hemoglobin less able to dissolve, and those molecules stick together when the oxygen level decreases. The molecules, because of this, clump into long fibers, and that gives that red blood cells with that molecule in it a distinctive shape, a kind of a crescent moon shape that looks like this. So that's a sickled red blood cell as opposed to a normal round red blood cell. Not only are they a different shape, but sickle-shaped cells are also more rigid than normal red blood cells. And because of their shape and because they're more rigid, they tend to get stuck in the capillaries. That cuts off circulation to certain areas of the body. Wherever that clog is happening, if we're not getting blood supply to an area of the body, that causes damages to cells, tissues, and even organs. That blockage happens in the brain, a person can have a stroke. Just going back to this, sickle cell disease is a co-dominant condition. People who have 2 normal genes or working genes will have all normal shaped red blood cells. People who are heterozygous will have some normal red blood cells and some sickle-shaped red blood cells. And people who are homozygous for sickle cell disease. So this is called sickle cell trait. And then people with 2 genes for sickle cells, all of their blood cells will be sickle-shaped. This is sickle cell disease. Cystic fibrosis is most common among people of European ancestry. Sickle cell disease is most common in people of African, specifically Northern African ancestry, right along the Mediterranean. Cystic fibrosis, on the other hand, is common among people of European ancestry. This is the deletion of just 3 bases. 3 of those genetic letters have been deleted in the big old gene for what's called the cystic fibrosis transmembrane conductance regulator, which we conveniently abbreviate CFTR. As a result, 1 amino acid is missing from the protein. So 3 bases, and we'll talk about this at the beginning of second semester, 3 bases code for a single amino acid. That 1 amino acid being gone, right here, causes the condition known as cystic fibrosis. CFTR, when it's working, allows chloride ions to pass across cell membranes. The loss of these bases removes 1 amino acid. Because that 1 amino acid is gone, the protein folds improperly. And because it's not recognized as being a working protein, it's then destroyed. So it's not found in the cell membrane anymore. It's eliminated from the cell membrane. You don't have to understand why, but chloride ions are important to get across the cell membrane. Now they can't do that. They can't get across. How does that affect the tissues throughout the body? There are digestive problems in the digestive system. They produce thick, heavy mucus that clogs their lungs and breathing passageways. The purpose of mucus is to trap foreign objects, dust, pollen, bacteria, viruses, and have it be brought back up by way of the cilia that line our trachea. Bring that back up to our throats where the cough reflex is initiated, and then we can cough that junk out. If the mucus is too thick, it stays there. It can't be moved up by the cilia, and it creates this perfect breeding ground then for bacteria and viruses. So we see a higher incidence of infection. People with 1 normal copy of the CF allele are heterozygous. They're unaffected. They're called carriers because they still, this working gene, this dominant gene, can still produce enough CFTR to get the chloride ions across. Two copies of the defective allele are needed to produce the disorder. So it's a recessive condition. This person is affected. Huntington's disease is a dominant allele. The allele for the disease that doesn't work properly has a long string of bases in which the codon CAG, coding for the amino acid glutamine, so again we have 3 genetic letters coding for 1 amino acid, repeats over and over and over again more than 40 times. It's not certain why these strings of repeats of glutamine cause the disease, but it just does. The symptoms of Huntington's disease include mental deterioration and uncontrollable movements. It's an adult onset disease. These symptoms appear in middle age. The greater the number of codon repeats, the earlier the disease appears and the more severe its symptoms. Remind me in class to talk about Huntington's disease and the story that I have about my aunt. It's interesting why disorders such as sickle cell disease and cystic fibrosis are still common in human populations if they cause so many problems. In the US, the sickle cell allele, that gene, is carried by 1 in 12 people of African ancestry. CF allele is roughly 1 in 25 people of European ancestry. The question is, why are they still around? Why haven't things been selected against them being around? Sometimes you see a genetic advantage to people who carry these alleles. Most African Americans today in the U.S. are descended from populations that lived in West Central Africa, where malaria is common. Malaria is mosquito-borne infection caused by a parasite, and that parasite lives inside red blood cells. Look at the connection between the incidence of malaria and the incidence of the sickle cell allele. You put those maps on top of one another, and you see a very close connection. Where you would see one, you see the other. People with one copy of the sickle cell allele are generally healthy, called carriers. Because they have these oddly shaped cells, red blood cells, they're resistant to the parasite. So it gives them an advantage against malaria. And again, like I said, you see this connection between the incidence of malaria and the location of where the sickle cell allele is found most commonly. So having the sickle cell allele as a carrier gives a person an advantage and an immunity or a resistance towards malaria. Now let's look at cystic fibrosis. In medieval Europe, more than 1,000 years ago, there were breakouts of typhoid fever. This is caused by a bacterium that enters through the body, or enters into the body through the digestive system. The protein produced by the CF allele, the non-working protein, helps block the entry of this bacterium. So individuals who are carriers, they're heterozygous for CF, would have had, again, an advantage when they're living in these medieval cities with poor sanitation and polluted water. Because they still carried a normal allele, they wouldn't have suffered from cystic fibrosis. Finally, chromosomal disorders. We've talked about these before as well last week. Non-disjunction we talked about. Not coming apart. In meiosis when homologous chromosomes fail to separate. Non-disjunction results in either extra or missing chromosomes, and that can lead to a number of chromosomal disorders. So we have extra chromosomes here and missing chromosomes here. If we end up with 3 copies of a particular chromosome, that's called a trisomy. Most common trisomy is called trisomy 21 or Down syndrome. Symptoms of that include mild to severe mental retardation, high frequency of heart defects, and others. Non-disjunction of the X chromosomes can lead to Turner syndrome. Turner syndrome only has 1 copy of the X chromosome. People who have Turner syndrome are not able to have children. Because they only have 1 X chromosome instead of 2, they don't go through puberty properly. Non-disjunction in males can cause Klinefelter syndrome, which is inheritance of an extra X chromosome. These individuals cannot reproduce either. There hasn't been any reported instances, as we talked about, of babies being born without an X chromosome. This chromosome contains genes that are vital to survival and development of the embryo.